Good afternoon. I am Dr. Sergio Arispe, and this lecture I'm going to be speaking on herbivore digestive anatomy. A brief overview of this lecture. First, I'm going to delve into comparative digestive anatomy between herbivores and carnivores. Then I'm going to look at herbivory and follow that up with different types of digestive systems. What's the anatomy of the digestive systems? What's the digestive end products? I'm really going to focus on foregut fermenters and also hindgut fermenters. So this figure on the top has a rabbit on the top left and a white-tailed deer on the top right, and these are two herbivores. Yet they're distinctly different. The rabbit is a non-ruminant herbivore. It has a simple stomach, so you see how small and simple this stomach is, and it has a relatively large cecum. The cecum is the long carrot-like projection pointing to the right. It's at the end of the small intestine and the beginning of the large intestine. On the other hand, the ruminant is not a simple stomached animal. It has a four-chamber stomach with a large rumen, and it also has long, small, and large intestines, as you can see in this particular diagram. The cecum isn't as important compared to that of the rabbit. So a lot of the fermentation that we're going to discuss later on this lecture go on in the stomach or in the rumen. Now down below we have two different types of, I guess we have an insectivore on the bottom left and a carnivore on the bottom right. The insectivore of this little mammal here has a very short intestine in general with no cecum. And the carnivore has a simple stomach with a very short intestine, colon, and a small cecum. This is because carnivores consume predominantly meat with some fat in it, and they try to pass that meat as fast as possible so they can stay nimble and quick. So this figure really highlights the nutrient cycling in ecosystems. As you know, plants are autotrophs, and they collect the solar energy and convert it into physical plant structures right here. And predominantly, they convert that into cellulose, hemicellulose, lignin. Those are the cell wall contents that Dr. Launchbow mentioned previously. And they also have starches, which are the cell contents. So this plant production right here and below ground is labeled primary production. Now herbivores will consume the energy right here in these plants. 50% of organic matter on Earth is tied up in cellulose in the plants. So this is a huge energy source for herbivores to tap into. And once herbivores consume these plants, in order to meet their nutritional requirements, they begin to gain weight, and that is secondary production, where these animals are gaining weight per unit area. So this is what we're going to really look at this particular lecture, is how herbivores access the energy in plants. The first thing I want to do is just look at a fraction of primary production that are consumed by various herbivores. In the grasslands, invertebrates consume about 10% of the vegetation. These are invertebrates like grasshoppers or crickets. Now, aside from that, nematodes, worms that are predominantly dwelling in the ground, consume 30 to 60% of the grasslands. That's an incredible amount of vegetation that the nematodes are consuming, and that's predominantly the roots and the decomposing plant structure of the grasses. Now we have the hoofed animals, or ungulates, on the African grasslands consume 30 to 60% of that vegetation type. And in the tundra, Lemmings consume anywhere between 20 to 90%. Birds can consume 0.17% of tall grass prairies. Their birds are predominantly granivores, eating the grain and seeds of these plants. And snow geese can consume up to 58% of salt marshes. So there are a lot of different types of herbivores, and they can have their own niche depending on the ecosystem. And so they can ultimately harvest the energy tied up in plants. So there are two structures I really want to hit on. The first structure is cellulose. And cellulose is a polysaccharide, or has multiple sugars, and each single unit is a glucose. And in particular, it's a beta D-glucose. There's 1, there's 2, 3, 4, 5, 6. And these are linked in what are called beta-1,4 linkages. Humans can't break that linkage. We can't access cellulose. And for that matter, neither can ruminants. However, ruminants or herbivores can or have a symbiotic relationship with microorganisms such as bacteria, protozoa, and fungi, which produce the enzymes necessary to break apart these glucose molecules from cellulose. And once they produce the enzyme cellulase, then the molecule cellobiose is produced. This is a cellobiose right here. It's a disaccharide or a 2-sugar compound. There's 1, there's 2. And that can be further broken down to beta D-glucose. So if you look at the top right, this is a graphical representation of what cellulose looks like. It really looks like sheets of paper. They're linear like a chain, and once those are compacted, the older the plants get, they mature, you have more cellulose, and the cellulose stacks up on top of one another like pieces of paper. Now, cellulose is a part of the cell wall contents. Now, within that, plant cells have cell contents. They have the starches. And this is a graphical or representation of starch. And there's 2 different types. One is amylose, and the other one is amylopectin. These are 2 different types of starch. Amylose has an alpha-1,4 linkage. These are individual glucose molecules, 1, 2, and there can be anywhere between 300 to 600 of glucoses here. So this could be one long string of amylose. And here's another glucose. So that's amylose. And then there's amylopectin. Amylopectin has a glucose molecule here, glucose molecule here, glucose molecule there, and has a alpha-1,6 linkage right here, meaning the number 1 carbon on this particular glucose connects to the 6th carbon on the other glucose. So these linkages aren't too important. I don't expect you to know them, but what I really want to point out is that starch and cellulose have different structures. And both of these, amylose and amylopectin, can be broken down by enzymes to make maltose, which is a disaccharide, a 2-sugar compound. This is 1 sugar, and this is the 2nd sugar, and ultimately broken down to glucose and alpha D-glucose. And look at the top right. Here, these starches can form, but they form a helical structure. It's different than the planar structure of cellulose. So what's the major purpose of the digestive system? It's really to provide for the assimilation of nutrients required for biological functions, including maintenance, growth, lactation, and reproduction. Now we're going to look at digestive systems. We're going to look at those of non-cellulose digesters, followed by hindgut fermenters, foregut fermenters, and what I want you to focus on is the different architecture of the gastrointestinal tract, the GIT the gastrointestinal tract. And look at the location, look at the size and shape, and compare and contrast the different digestive systems of each of the different animals. Then really focus on the nature and location of the digestive agents. I sum it up for you in tables, so I really want you to focus on that. Are there enzymes involved? Are there digestive juices? Is there fermentation? Lastly, know the chemical nature of the end products of digestion. Are they amino acids, volatile fatty acids, monoacylglycerol, free fatty acids? And once again, I try to sum this up in a table after I highlight the different type of herbivore. So this is an overview of the gastrointestinal tract. It starts with the mouth and ultimately ends with the anus. Everything in between is the gastrointestinal tract. So unfortunately this slide didn't turn out well, and I apologize for that. But what we can see here is what's thought to have occurred from an evolutionary perspective. In the Cretaceous period, the dinosaurs were said to have gone extinct, and that's about 65 million years before the present. Now, on the x-axis, the left three labels are the dicots, monocots, and grasses. And the dicots were the first plant materials to evolve in the Cretaceous period. And that corresponds with the letter B, and that is the common ancestor between simple stomach animals and ruminants. So these animals consumed dicots and monocots, and these two plant types are high in cell contents, and low in cell wall content. So they have less lignin, less cellulose, and less hemicellulose compared to the grasses, for example. So these animals that first evolved had simple stomachs because they were able to pound out the cell contents and utilize the starches from those plants. So this is a figure of a pig, digestive tract. Pigs have simple stomachs, as you can see right here. There's nothing elaborate about the stomach. They're called monogastrics. That means one stomach or one gastric stomach. And examples are humans, reptiles, and swine. And these species derive energy from starches. And cellulose is not a primary energy source. So there's limited fermentation going on when we consume plant products. Next are cellulose digesters. One example is the horse on the left-hand side, which is a hindgut fermenter, and on the right is the cow, which is a foregut fermenter. So let's go to hindgut fermenters first. Once these animals consume food, they pass it along to the esophagus and then ultimately drop it into the stomach. And in the stomach, there's some activity, hydrochloric acid is produced, and it begins to denature some of the proteins. Those proteins are further broken down by the enzyme pepsin. So pepsin will break down that protein to peptides and maybe some amino acids. From there, the ingesta, everything that's been consumed, passes from the stomach now into the small intestine, and that's where absorption, a lot of nutrient absorption occurs. And then afterwards, what's not absorbed passes to the large intestine. And then you have two different types of hindgut fermenters. One are Cecal fermenters, such as the rabbit and the guinea pig. And the other two are colon fermenters, like the rhinoceros and the horse. And ultimately, after fermentation occurs in the large intestine, the animal passes the feces through the. So, cecal fermenters. The rabbit is a typical example of a cecal fermenter. And as you can see in this particular diagram on the left-hand side, they have the simple stomach with a long small intestine. And then at the end of the small intestine, there's a cecum. It's called the blind pouch or the blind pocket. There's one way in and one way out. It's the same hole. So it's, in essence, a pouch. And there's a lot of microbial organisms, a lot of bacteria that live in the cecum. And this is a primary site of fermentation. And these microorganisms produce the enzymes necessary to break down forage. Once that forage is broken down, the animal then passes it to the large intestine, where ultimately it's excreted. So on the right-hand side, you can kind of grasp more or less the size of the cecum. But it's the next slide that really shows the capacity of the cecum compared to the rest of the gastrointestinal tract. The cecum can hold up to 100 to 120 grams of the ingesta. That's 10 times more than the stomach, which can hold about 10 grams, maybe even 30 grams. And the stomach, which can hold up to 90 grams. So the cecum is a place where fermentation occurs predominantly in rabbits, guinea pigs, also the rat. And then another example of that is the koala. These are all hindgut fermenters where they digest or they break down the food in the cecum. Now, in the case of the rabbit, it's interesting that once the food goes through the digestive tract, sometimes the rabbit expels feces that it consumes. Now, there's two types of rabbit feces, and some of you may have rabbits of your own and may have seen two different types of feces. One is a hard cellulose-containing perfect ball that typically falls to the ground. Now, that has predominantly cellulose. Now, the second type of feces is excreted, and it's a gelatinous feces that stays around the anus. And sometimes the rabbit will reach back and consume that second type of excrement and consume it, and that process is called coprophagy. Coprophagy. Now, there's a second type of hindgut fermenter, which are colon fermenters, and these are animals such as the horse, the elephant, the rhinoceros. And look at the bottom diagram, and these animals are simple stomach animals, the monogastrics. But you look at the anatomy, it has a long small intestine, but look at this large intestine. The cecum is fairly large, but it holds no weight compared to the extremely long, large colon in the horse. Now, to give you a perspective on the capacity of this GIT, look at the picture on the top right. The stomach can hold anywhere between 8 and 19 quarts, and the cecum can hold 28 to 36 quarts. But if you look at the large colon, you can see that the large colon holds 86 quarts, and the small colon can hold up to 16 quarts. So, due to that size, there has to be something that's going on, and indeed, that is where microbial fermentation goes on. The microbes are able to produce the enzymes necessary to break down cellulose, and that cellulose ultimately provides energy for a type of energy for these animals. So, in summary, for hindgut fermenters, the stomach is able to break down protein. Protein is the substrate. Now, it does that by producing the digestive agent, or the enzyme pepsin. And ultimately, what's produced are amino acids or peptides. So be sure to write in peptides right there as an end product. Now, in the small intestine of hindgut fermenters, carbohydrates are broken down, or carbohydrates, proteins, and lipids are all broken down by pancreatic enzymes. And these pancreatic enzymes include amylase and glucosidase, which break down starches, right, the carbohydrate starches, to glucose and maltose. Now, the small intestine, there's also the pancreatic enzymes that are breaking down these proteins. Those enzymes are trypsin and carboxypeptidase, and they break down proteins to peptides and amino acids. And lastly, the lipids, or fats, in the form of triglycerides or triglycerols, excuse me, are broken down to monosaccharides or free fatty acids. And last but not least, the cellulose is a complex carbohydrate that the microbes are able to start breaking down, and they convert the cellulose, or ultimately the molecule that's freed as glucose, they break that down into volatile fatty acids. The large intestine is also a site where mineral and water absorption occur. So that's a summary of the hindgut. And next, I want to highlight the evolution of foregut fermenters. And B was the common ancestor between simple stomached animals and ruminants. And so is A, which evolved in the Paleocene time period, gives rise to, it says hippos and pigs. So pigs and hippos. Those are simple stomached animals. And then it gave rise to the tylopods, which are llamas, alpacas, vicuñas, guanacos. Those are New World camelids. The triagelids, which are small deer, and cervids, which are your larger deer. And it's about this time in the Miocene period where grasses began to evolve. So grasses began to evolve 26 million years before the present. And what you see is ultimately the evolution of the giraffes, the antilocaprids, which are antelope, and the bovids, or your grazers, your cattle. And cattle are thought to have evolved after grasses evolved. So that's important because cattle are able to digest the grasses that maybe some of the other animal species weren't able to digest. So it's thought that they evolved once the grasses arrived 26 million years before. So this is the overview of the gastrointestinal tract in general. And what I really want to focus on is the stomach. And the stomach is a place of fermentation for fermenters. Now, one example of a foregut fermenter is a kangaroo. As you can see in this picture, the kangaroo is eating grass, and indeed they're able to ferment that grass in their stomachs. So if you look to the right at the digestive tract, you can see how this stomach is pretty complex, and it's broken up into three sections. One section you have which is the top half. You have microbial fermentation going on. That's where you have the coarse forage that the animal consumed. Then the second third of the stomach is once that that forage is broken down by the microbial organisms, by the bacteria in the stomach. It then passes to the next section, and ultimately to the third section, which is the bulb-like structure just before the small intestine. And that's where gastric acids, that's where hydrochloric acid is produced to break down proteins to peptides. Now, unlike the rabbit, the kangaroo has a very small cecum because the fermentation is predominantly going on before the small intestine. So that's one of the things that should have stood out in this particular picture. Now, another type of foregut fermenter is a ruminant. And examples of ruminants are cattle, sheep, goats, elk, white-tailed deer. And the ruminant has 4 compartments to its stomach. And in order to learn a little bit more about the ruminant, what I want you to do is click on this YouTube video or input the YouTube video into your favorite web browser and watch the video. It discusses the anatomy of the ruminant and a little bit about the different compartments of the stomach. So if you can watch that and then return to this presentation, then that would be great. But in general, ruminants have very long small intestines and large intestines, and their stomach has 4 compartments: the rumen, reticulum, omasum, and abomasum. And it's the reticulum and the rumen where the majority of the microbial fermentation is occurring. That's occurring with bacteria, with protozoa, and with fungi. And it's said that in 1 milliliter of rumen fluid, you have over 1 billion microorganisms floating around in that stomach juice. So I'm going to highlight those 4 compartments later on, but I just wanted to briefly touch that here. So once the ruminant apprehends feed, they begin to chew, they begin to salivate, and ultimately swallow that food, and it goes into the esophagus. Now, if the animal's nursing and is taking in milk, the esophagus, the hole that leaves the esophagus, will ultimately be pressed up against the omasum, which is the 3rd compartment of the stomach, via the esophageal groove. So via the esophageal groove, you follow the arrow down, and the milk goes into the omasum. From the omasum, it goes to the abomasum, and then to the rest of the gastrointestinal tract. So now, why does the esophageal groove put milk into the omasum? Why doesn't milk go into the rumen or the reticulum? Well, one of the reasons is that that's the site of microbial fermentation, and microbes would go crazy and multiply and begin to produce toxins, and ultimately give the calf or the young ruminant a bad set of diarrhea. So it's only in suckling animals that they have that esophageal or ruminal-reticular groove to pass the milk so it doesn't ferment in the reticular rumen. Now, if these animals are more mature, the food's going to be passed from the esophagus into the anterior rumen, or the reticulum, which is essentially a pouch that's attached to the rumen. There, the food is mixed. The microorganisms, the bacteria, fungi, and the protozoa really break down those cell contents and the cell wall contents and ultimately produce volatile fatty acids. Those volatile fatty acids are 1, butyrate, that's butyrate, 2, propionic acid, and 3, acetic acid. So those volatile fatty acids are absorbed in the rumen. Now that leaves some fiber, some food still churning, and that, once it breaks down into smaller particles, it goes from the reticulum to the third compartment of the stomach called the omasum, and from the omasum it goes to the fourth compartment called the abomasum, and ultimately is left on the ground as. So in the top right you can see how large the rumen is in the ruminant stomach, and the major purpose of the rumen is to be a site of anaerobic fermentation. It's also a place where volatile fatty acids can be absorbed, and ultimately where contents can be mixed. The picture on the bottom is what the mucosa looks like. It's filiform and foliate. They're finger-like projections which increase the surface area in the rumen so that the animal can absorb a lot of volatile fatty acids. Now, ruminants and foregut fermenters can use volatile fatty acids for energy because they have the enzymes or the chemicals necessary to convert that to energy, whereas humans or pigs or monogastrics don't. So the next compartment of the stomach is the reticulum, and the reticulum distributes feed to the omasum, which is the third compartment, and this is the origin of primary rumen contractions, and you have some VFA absorption here. Now this mucosa looks like a honeycomb, and it's like that in order to have an increased surface area so volatile fatty acids can be absorbed, and ultimately it traps small particles. And once particles get small enough, they pass from the reticulum into the omasum, which is the third compartment highlighted in green on the top right hand side of the screen. The purpose of the omasum is to pump food to the abomasum, which is the fourth compartment. In addition to that, there's active exchange of sodium and potassium, and also some volatile fatty acid and water absorption. So ultimately, the omasum is regulating flow into the abomasum, and the mucosas are arranged on muscular laminae. These are like pages of a book, they say, and it helps regulate, once again, food going from the omasum to that. And the last compartment of the ruminant stomach is the abomasum, and this is the true gastric stomach. It produces hydrochloric acid, and it also secretes proteolytic enzymes. The pH in the abomasum is between 2 and 3.5, and what that does is that will help prepare food for the lower gastrointestinal tract. And the abomasum is arranged in folds. So to this point, up until this point, I've highlighted the purpose and also a bit about the mucosal structure of each of the compartments in the ruminant. So I really want you to be able to know the distinct differences of those compartments. So yeah, you can ultimately understand the nutrition in later lectures. This is an overview, a summary of the substrates, digestive agents, and end products in the ruminant. And what I really want to highlight is that the microbes here in the reticular rumen are able to break down the protein, carbohydrate, and lipid substrates into microbial protein, volatile fatty acids, and ultimately free fatty acids and triacylglycerol. So this is the predominant difference between simple stomach animals, monogastrics, which include hindgut fermenters, and ruminants and foregut fermenters. So The last thing I want to point out here is that the size of the stomach shifts in ruminants when they are born compared to at maturity. And at birth, the rumen is approximately 25% of the total stomach capacity, whereas the abomasum is a good 60%. Now that changes as the ruminant begins to mature. The rumen takes the primary role in breaking down food, and that can reach up to 80% of the stomach capacity, whereas the abomasum has a different role in it, but it gets smaller relative to the rest of the stomach at about 8% capacity. So ultimately, in summary, the architecture of the GIT and the agents of digestion are of utmost importance to determine digestive function. Next, the ruminants and hindgut fermenters have well-developed fermentation sites which allow them to digest cellulose. It's actually those microorganisms that the animals have a symbiotic relationship with that can help them break down and utilize that source for food. Third, hindgut fermenters have greater rate of passage and can survive on lower quality food. So hindgut fermenters pass food and they don't retain it as long as ruminants do. And lastly, ruminants can eat lower quality forage but must retain the food for a longer period of time. That way, the microorganisms in their stomach can churn and really digest and break down those carbohydrate structures so the animal can ultimately use it for energy. So that's a wrap up on this lecture. So if you have any questions, feel free to email me, and my email address is aris3391@vandals.uidaho.edu.